So basically this is a paper, right? Nature Communication. So we did like last year, and that was like 7,000 genes and then so many siRNAs, right? Because on an average 3 siRNA targeting one particular gene, right? So what we did is we extracted all the imaging features, first order statistics, second order statistics. So that is 53BP1, all the mitotic DNA damage marker. So 53BP1 and gamma H2X, right? So they all represent on an image as a foci within a DAPI nuclei. So first we also segmented that as a foci, and we counted how many foci of 53BP1 and gamma H2X are there in each cell for each siRNA for each stratified by different genes. Okay, so you can imagine the size of the Excel file, right? And there were basically 3 replicates for every planes. Okay, so for every siRNA 3 times the same exponent. So the number of channels, one for DAPI for DNA, right? And then EDU for cell proliferation marker, and 53BP1 and gamma H2X. They both are for DNA damage control marker. Okay, and then we did some kind of a machine learning stuff, but that's a different part. But what we did is basically for every cell you get a probability of whether or not, or how much likelihood that cell has a morphology which corresponds to 53BP1 damage or gamma H2X or EDU, right? So every cell will get a 3 probabilities basically from 0 to 1. If the cell is very similar to 53BP1 phenotype, then it would get more like 90% of 53BP1, 70% of gamma H2X, and 50% of EDU, for example. If the cells are proliferating at that particular moment, then it's more kind of, right? So it's like a 3-dimension we have. And of course DAPI as a single in order to stratify whether or not they belong to S phase, G1 phase or G2 phase. Okay. So this is the file I can show you here. By the way, one of the plot is just a normal table, right? So if you just click on table, it is as same as you are looking at in Excel file, right? So this will show me that what kind of data I have here. So I have a row column gene belonging to gene name and then siRNAs. Right. This is very interesting because we have done all this analysis in MATLAB, which is not using scanner. So there's a possibility of saving individual cell that we've segmented as a snapshot, and this is just the location of that particular image. So I have basically cell image for DAPI, cell image for 53BP1 channel, and likewise for EDU and H2X. That's the column basically, and the column represents the location in which that cell is stored, right? And this is what I was talking about. So every cell, every row is basically a cell for particular gene and siRNAs. It gets 3 votes: 53BP1, EDU, gamma H2X. Okay. Now, whole idea is basically I want to pose a lot of questions, biological questions, and these people have tried doing this, which I hardly understand, but I could tell you that okay, if x-axis, let's say for this particular figure, probability of mitotic aberrations, so that's basically gamma H2X. Okay, probability of being a gamma H2X. Y-axis 53BP1 foci, and these are the scatter plot, right? And every scatter plot is actually representing a particular gene, right? And as you know that there are multiple siRNA targeting for one particular gene, so you get basically aggregate vote. So let's say if there are 3 siRNAs, right, then for one particular gene, it gets 3 votes from 3 siRNAs for one particular cell, right? And this is what that value is basically. Yes, so that's what I'm going to show you. How did I do that? So basically, but I get this XLS file after MATLAB and all kind of fancy image analysis stuff, but this is what you get basically. Now, how to represent and how to pose different questions, right? Let's go to this scatter plot. Just click here, scatter plot, right? So by default, it has done some kind of a judgment. Also, the first case. So remove the color bar x-axis, put gamma, vote H2AX, right? Imagine this is just the probability 0 to 1, and then here 53 BP1. So now when I say marker by row number, meaning that it's data for every cell now, for all the sRNA, for all the genes, and for all the position and likewise. But maybe I'm not interested in cell level. Maybe I'm interested in only what is happening actually on the gene level. You see, these are the genes now. I can also go for, okay, I'm interested in what is happening in siRNA level. You understanding now? Your x and y axis is the criteria that you see it, but the moment you say siRNAs, you have to aggregate the vote, right? Because for one siRNA, there are multiple cells. Now, how do you want, basically, Spotfire is asking you, how do you want to aggregate this many votes, right? Because there are multiple cells within one sRNAs. How do you want to aggregate them and show it as a spot on the box plot, spot on the scatter plot? And this is what this aggregation is basically, right? So what I want is that they take all the cells which are represented by particular siRNAs here and give me an average vote for that one. So that's what I'm changing this aggregation now to average. Okay. Similarly, I have to do it for 53 BP1 and then average. Now it looks good. Again, it goes back to the same scale, 0 to 1, 0 to 1. But now they are stratified by sRNAs and aggregated by average value. But where do you see there are 700? No, actually I cropped it. Sorry, I cropped it. Okay, because if I go for this one, of course it will take some time, right? I remember I cropped it because that you can see it from this random number, because I have done this random number and cropped it basically. Yeah, because that was just for demonstration. Okay, where was it? Yes, here. That was good point. Yes, and then so this is just the scatter plot, right? Average value stratified this one. I can also average based on gene level. Again, it's reduced, right? Because genes are less than the sRNAs. So I can right click, go to properties, make it more smarter. So I go to labels, and then maybe I want to display the gene name. But how do I want to display it? Just say most common. Then you have a choice of displaying all of them. So by default it will display all of them, or marked rows. What is the difference? The difference is, if it is in marked rows, then it will only display to those which I have marked. Otherwise, if you want to display all of them, just go back to formatting. Where was it? Labels, and then you say all of them. Now they're displayed. Okay, so you can see, okay, I think there is something interesting going on with this particular gene, right? That its gamma H2AX is more and the 53 BP1 is less. Well, normally they should correlate, right? I mean, they both are DNA mitotic damage, but it depends on the morphology, and there are so many other factors on which this the vote has been evaluated. So that's why it's very interesting, right? So, and you can also see the negative control. They are also extreme, right? So they are basically more gamma H2AX, like average, more than average, and then also the 53 BP1 is also high here. Okay. But the interesting point here is now that maybe I'm interested in only looking at this particular gene, and I want to know what is happening to the sRNAs with its respective sRNAs. Okay, so for that, what Spotfire can give you is that if you just right click here and say create details visualization. Okay, so if you click this and go to again this scatter plot, it will create a scatter plot which will be empty, but that depends on which points you mark it. Okay, that only corresponds to that particular thing. So if I mark it here, it will give me something this too. See, and I say mark by siRNA now. So now I know that for this particular gene, there is only one siRNAs. Because now it is a marker by siRNA. Okay, but maybe there are some particular genes which may have multiple siRNAs. You see here? Okay, there are two siRNAs. Okay.
Was the aggregation of the data, for example, for the things that you have 3 siRNAs?
Yes.
Did you do this in your analysis? Is it a MATLAB thing, or does it offer it already?
Sorry, come again.
You put together, let's say, the data from the LR, whatever, the two siRNAs.
Yes.
You did your analysis with MATLAB already?
With MATLAB, yes.
Okay. So this predates
Yeah, exactly. Yes, exactly. Because I have been given the labels to individual wells, right? Yeah. An individual well is siRNA, right? So I have segmented, extracted all the features, and then given that feature label as what they have given me, right? Okay, but then you can actually see it, right? So that's basically details on visualization, right? So you select it, and then you get information about what kind of siRNA they have it, right? You can actually change it to change this visualization to table just to see actually what's happening. So if you select this one, this guy over here, then you know only rows of that particular gene will be displayed here, right? Isn't it interesting? Just to basically, or maybe you just go and right click and go to, let's say, a bar chart. Okay, in the bar chart you will say you're interested in knowing what is the BB1 foci intensity, average intensity for that particular gene. So you just go in. This is the basic intensity. And if you have multiple one, then of course give multiple. See. Okay. And you can add multiple information. So it's a gene, then you go for the siRNA, right? If you select this one here, then it's siRNA IDs. So that's basically a gene which is FAM178A and which has these 3 targets siRNAs, and that's their value, basically average vote value. Okay. Again, if I had given position and the cell ID, I would have given you actually exactly the pinpoint number of the for one particular cell. So many cells corresponds to one position, right? Many position comes to one well, and every well represents one siRNA, and there are multiple siRNA for one particular gene. So imagine the hierarchy, right? But that is possible. Everything is possible here in Spotfire. Okay. Yeah, that's basically one feature of details on visualization. Then maybe you're interested in knowing. Okay, this is very important point. Right click, properties, go to labels, and maybe you're interested in marked rows, but not the value, but maybe the images. So how the DAPI would look like. Let's say the most common. Go there. Then in this, so how do you want to display it? You want to display it as a text? Okay, no. I want to display it as an image from the URL, because you remember that one of the column was the location in which the cell was segmented, right? I'm not sure whether it will work because I'm actually reading it from the microscopy data server, so maybe there is some problem, but let's try. Let's try what happens. So if I select now, oh, it's working. Good. You see now? So you can select it and see, oh, that's nice. Okay, there's something, what's interesting about maybe, and I think we should also display the tooltip. So in the tooltip, if you go tooltip, tooltip, you can also say add. I'm also interested in knowing what gene they have. It's the first gene, that's fine. And also I'm interested in knowing what siRNA they belong to. siRNA ID. Okay. And maybe I'm not interested in this one. So basically if you put it here, put cursor over here, so it shows me all the siRNAs, all this information. Got it? What else? This gene preview, and similarly you can actually change it for other channels also, and you can have multiple such things. So if you go right click, properties. Labels you can have only one image, right? Because if you go to the labels, for example, here there is no plus sign, so you can only select one criteria that you want to labelize it. But if you want multiple one, then you can go to Tooltip, and instead of this one, you can also add the image. So Tapi as an URL, okay, and one more. Go to 53BP1. So basically, I have all information about both the data and the image for the description. See here? You know what's happening actually here. There's a representative class. Okay. I can imagine why the 53BP1 is more here. You see here? And gamma H2X, because you can see from the image itself. Yeah. Right. Of course, it's less here. It's also, that's a DP basically. That's a false positives. Okay, so there are so many things you can do it. Now let's add. So now they both are mitotic damage indicator, but maybe I'm interested in stratifying them based on the cell proliferation. That is the EDU marker, right? So the size by EDU. Let's go for EDU vote EDU, and go for the average one. Yeah, they're not that much changing, but that's fine. Then go to properties again. Go to labels, and labels I would say like genes. Oops, right click, labels, and I want to display all of them. I don't want to mark it. Yeah. Now you can see actually the biggest one is this one. Like the biggest diameter means that the cells within this genes are basically proliferating a lot. Maybe something that you have to investigate further. So that's possible. Negative control, average size, maybe something is interesting there. Here. So likewise, so that's all questions that you can actually pose, and they are actually showing it here. So you can see here that the size of EDU, that's based on the size of the spots. X-axis is your H2X, gamma H2X, Y-axis is the BP1, and this is what they are showing. Now what? Yes.
The beats are arbitrary, so
the beat. So yeah, exactly. The size you can control that. That's possible.
So you can set, for example,
yes.
Now it's 443.
Right. So you go to properties. And automatic. So value based. You can give value. Yeah.
But do you know based on what it's calculating? It's an average, or it just randomly assigns numbers?
No. Basically, it goes from minimum to maximum.
Okay.
Right, and then just divides into let's say quartiles, intervals.
Okay.
Yeah. I don't know exactly what criteria they use, but that's like a manual. Yeah.
But you can set it manually.
Oh yeah, exactly. Yes, that's possible. And maybe you can go by color, right? So color may make more sense. So let's go for EDU. Vote EDU, average, average value, and go for the auto P. Right, methods, and maybe I don't need a size anymore. So basically, the X, this color, well, this is really bad actually. More colors are confusing, right? So I will reduce it to 2, but then I know option, right? So all the greens are basically, no, the blue ones are more cell proliferating. So, oh no, the other way around, the greens one, they are more than 0.4, and that criteria you can change it, right? Maybe it should be 0.5 here. That's a 50%. Okay. But now you got it, right? And then you can actually use a shape as an information about, let's say, Tapi.
Here it's only based on one cell and one image. Or
this is one cell, one image. So every feature is based on, it's a cell-based feature. We haven't aggregated. Now we are aggregating actually in Spotfire, but the file here, file I showed you, this is, every row is basically a cell.
because you always only were shown one cell, right?
Yes. Yeah. Exactly. Yeah. Of course, for showing cell Spotfire will ask you, because it cannot show you all the cells, right, for that particular genes. That's not possible. It's only one image. That's a limitation of Spotfire. So you have to give what criteria do you want to see. Let's say if I have, let's say I have 100 cells for one siRNase. Is it the first or most common or something like that? That's why I used first actually, because that should be enough as a representative for an siRNA. Well, you have to see the variability in the cell also. But yeah, okay. So that's all about the Spotfire over this one. There is one more thing that you can do. Let's say, for example, especially for the DanStem and also for CPR second flow, that people do live cell imaging, but for 2D, and for you guys you do 3D. So if you do live cell tracking using Imaris, and then you export your data, one of the columns is the time, and then the feature would be according to your interest. It may be cyclic even degradation stuff, CPR. Maybe for you it's just the GFP expression, like fusing and all. But no matter what you have one column which is time, another column is some feature that you are interested in. Right? In such cases, you can just click on this line chart. Click on line chart. X-axis is your time. Y-axis, for example, in this case, I have a cell count. So maybe the cell counts are increasing basically over time. Something is happening. And this is what you can see, the profile basically. Color by, so if you have multiple such criteria, you can have multiple such line plots. Right now, I just have basically a cell count as only a feature. I don't have any other feature. But if I had like a multiple columns, which also varies with respect to time, I could see multiple line charts on the same plot, and then I can compare it. Got it? That's also possible. That's all about line chart. Okay. What else? Any question about the spot actually? The scatter plot. The scatter plot is something that you're gonna use it a lot. That's also probability of mitotic aberrations. That's also scatter plot. You see, and most of the time you will display as a scatter plot. There are something like, for example, the clustering thing, right? Maybe you want to actually do not want to use it as a bins to give a color, but you want to use a cluster. That is not possible in Spotfire. So what you have to do is you have to do some kind of MATLAB script, and you have to generate a column which would tell what cluster they belong to, like particular number of cells, and then you come back to Spotfire and then use color by cluster class, and then you will see nice clusters. Okay. I wanted to give you this example. This is clear, right? Maybe this one I go. Right, you can change actually. Either you can click to scatter plot, or if you do not want bar chart, then you just right click and say change visualization to scatter plot. That's also possible. So directly goes there. Now what I want is here, I want this to be total intensity of the API. This one, mean intensity of the API. Marker bar. And then color by, because when I did this analysis in Scanner, I have already created a gate. Okay, gate meaning that G1, whether or not the cell belongs to G1 phase, G2 phase, or S phase, and mitotic phase. Okay, and that's why you see this as a one of the column also in this case. So if I say color by G1, it will give me those cells which are actually in the G1 phase. Then I would add one more. G2, that's basically here, right? That's what I was talking about before. And I think this cell should belong to the mitotic one, right? Because their mean intensity is higher, and also they have the average kind of the intensity. Okay, let's see actually. add more and say mitotic. You see, there's a mitotic here. That's mitotic. This is according to, you see the m is equal to 1 and the rest are zeros. Right. That's just like a second validation. So basically you can do it either in scanner, or you can just do your segmentation in MATLAB or scanner and directly come here and then you yourself make it like this kind of a marking by yourself, and then you can give the naming. Naming, I will tell you how to labelize it later. But this is how you actually visualize the data. Okay. So about that. What else? Bar chart we have seen. Oh yeah, by the way, in this case, for this particular study of siRNAs, maybe you are interested in knowing what is happening actually for what is the average value of the vote of the 53BP1 for one particular siRNAs, for one particular gene, right? But you do not want to look at it as a visual perspective, but you want a number. Maybe you want to display or you want to do some kind of post processing. In that case, this is useful, which is called as a cross table. Okay, the cross table, if you click on cross table, there are 3 dimensions that you can control. We have seen by default X and Y, but there is also another dimension, which is Z dimension, right? So in Y-axis, I am interested in looking at genes, and not for genes but also for every siRNA here. And what I want to look at, actually I am interested in looking at what is the average vote of the 53BP1 for a particular cell. And then here I would say, okay, I'm not interested, I should say remove it, right? So you're going to see only the one column, right? So this is a gene. This gene has 2 siRNAs of that particular name, and every siRNAs may have multiple cells, right? And this is the average value of all these cells, which is basically this one, right? And with this I can actually sort it. I can sort this, but I can also add another one. Maybe I'm interested in the vote of EDU, average vote EDU, and also the gamma H2X, right? Not the count, but the average one. Oh, not the PV, sorry. So that's gamma H2X. Gamma H2X. Okay. That's the average one. Okay. So I have all these features basically. So I know what is happening now here. And you can actually sort it, right? So the maximum is basically it may not work because now we have basically 2 criteria in the y-axis. So if you just say maybe I'm not interested in genes, I will see it later. But for every siRNA I'm interested in this one, then you can actually sort it. See, so the maximum vote for gamma H2X is for this particular siRNA. And then you can actually go back and say, okay, add the genes. The other way around, right? But then you can highlight that particular, so you can click it, press Control, and then that part will be in the green color. Okay. Do you want to do it again? Go to gene here, remove it. This was the topmost. You see, this is selected here. That's highlighted. So I know a priori that this is basically this one, TCC C9. There is something interesting with this one, which has the maximum gamma H2X average. And then I know what gene they belong to because I have already displayed here. That's a gene. Okay. What else I can do here? That's all about the cross table. By the way, you can export it, this one. So just file, export, data to file. Then you say, do you want to export this visualization? Yes, I want to export visualization data for all the items. Yes, okay. and then you can save it as a text file or XML file. If you want to do some further analysis on this one, that's possible. Maybe you are interested in actually calculating the average of these three guys, all these three together, three words, I mean the three morphology criteria basically. Then how do I do this? Just right click and you say grand total for the rows. That's basically total rows. This doesn't make sense. Control T and subtotal for count all. That's if you put gene over here. Right, you can see subtotal for genes, and it gives you a subtotal. Should give you a subtotal for gene. Yeah, that is subtotal. See. So for every gene, it is giving you the total of all the siRNA. So this is basically all numerical values, and this is what we have done actually in the pictorial. Right? When we say marker by and stuff like that, and how do you want to aggregate that? Okay. Okay, this is interesting. Combination chart. Okay, so nobody has used it, but I can tell you in some scenario where you would like to use this. So what is a combination chart? Combination chart is nothing but the bar chart and the line chart. That's a combination. So you can have both possibility in one plot. Now, where do you want to see it? So maybe let's say these are the genes basically. And what is my criteria of the gene? Let's say my gene criteria is the average vote of 53 BP1 in that gene. Go for average. It's my average value. But I also want to plot the EDU as an average value, as a line plot for every gene. So click here and go to EDU vote, average value. Okay, now it's basically side by side, but you just go properties, go to series, and in this EDU you say instead of bar, I want to have a line. That's line now. Then you go to labels, formatting, and formatting you say show the line marker. Okay, that's my line marker. Right. And say break lines on draw as a state, so you can actually like a state also. But I think I like this one. It's more continuous. Right? That way you can actually control and see it actually. The two information on top of each other. And you can have a multiple such stuff. So you can have a multiple lines, multiple bar charts in one plot. Okay, this is all about the combination part now. The combination part here. Pie chart. This is very simple, right? And everybody knows what is pie chart. Pie chart is basically you can stratify. Now, for example, this is a gene, so that's color by gene. But then you can actually use some criteria. So it's if you go here, right click, properties, data, it's fine, appearance, sort by sector, size, font, size. So in the size criteria, on what basis you want to divide your pie chart, that you can control. So that will be based on, let's say, not the vote, but for 53 BP1 and the average value. Just to see how they organize. I think they're equally likely in that way, right? This is not a good example for pie chart, okay? But you imagine, right? I think the best example would be that region example that I gave you, right? The Pacific region, Europe, and Asia, and stuff like that. Because this is the highest in hierarchy, so it should be basically kind of a well, for example, right? Within the plate. Sector size. That's fine. That's a pie chart. Then the second one, scatter plot we have seen. A parallel coordinate plot. The This is also interesting. Now, in this case, what is this? The parallel plot will tell you. It is basically without doing some kind of machine learning. If you want to find a similarity amongst a particular genes or group of genes based on some criteria, then this is how you should do. Okay. So first, for that, you just click on this parallel plot. Right click, properties, go to columns, and in the columns you have to select on which criteria you would like to evaluate that similarity of the genes. Okay, so of course it cannot be a gene, cannot be a row, cannot be any one of these. It should be the numerical value. So let's say vote of 53pp1, vote of edu, and the vote of gamma h2x. And then color by gene, well, I don't care, remove it. This becomes so ugly, right? Because they have never thought of such kind of example. Basically, this is good for if you have a store. This is for the purchase and sell stuff, like for the marketing stuff, right? Maybe if you have some criteria, imagine that you want to buy a TV, and there are 3 criteria for the TV. For many vendors, and in the y-axis there are basically different vendors, and every TV will have these 3 criteria. Maybe what is the picture quality, what is the sound quality, and what is the cost. And there are so many such lines would be there, right, in the market. Now, depending on what is your criteria, maybe you want a high picture quality, high sound quality, and less price. So you can actually group them likewise. You understand my point? In this case, it is becoming too much because there are so many such SRMAs here, right? So it's line by now row number. Let's go for G. Now it's good. So for every gene now, it is calculating the sum of 53pp1, sum of edu, sum of gamma h2x, and now you yourself from your eyes, you have to see the similarity. Do you find some similarity in this case? And by the way, for every criteria, everything is relative. You see 100%, there is always one thing which is 100%. So for all genes, the scale is relative. Yeah, 100% to 0% for every criteria for all the genes. And maybe actually I would say these guys you see here, they are different actually, because they are basically going in this profile and then going up and up. Maybe there is something interesting in that. So just select that and see what these genes are without even calculating this linear discriminant analysis and stuff like that, or principal component analysis. This is just, it's very simple. By the way, nobody has used it so far, but there is a possibility of using it without doing some fancy machine learning stuff. And then you see what kind of, these are the guys actually. They are kind of similar. Not on this 2 criteria, because right now we are only looking at 2 criteria. But right now we are looking at 3 criteria now. And we can have it as many criteria as we want in this case. So I think there is something similar with this particular family of genes. So let's go and investigate. You understand this part? The final part is the box plot, but before that, what is this? Graphical table. Well, graphical table is hardly you would use it, but let's see. The graphical table, it's basically a table, but within the table, instead of just having a numerical value, you can have pictures, or you can have a line chart and things like that. That's a graphical table basically. Instead of numerical value, you can have a picture, you can have a thumbnails and things like that. So now you have an x-axis here, Sparkline. Right click, properties, go to appearance, font axis. To be honest, I never used it actually. This thing. Data. Okay. So you can actually add, so there are 4 possibilities of calculating and displaying it apart from numerical value. One is a Sparkline, meaning that if you have a timeline data, then you can actually show it as a snapshot, just like a small time point, like a line chart, as a row value, as a picture, calculated value, icon, if you have an icon, or the bullet graph. Right. So let's go for the icon, and then icon you have to make a rule. Rule would be if value, what was my value? Maybe let's say top value. Shape is this one. Okay. But based on which criteria? Based on, let's say, vote of 53 pp1. Right. And then the average value. That's all my top one. That's you see here now, stars are coming up. So that stars is governed by the rules that we have created. And likewise, we can have multiple such symbols. So just by looking at the this graphical table, one can know which genes are expressing more 53pp1 or more, and which genes they are responsible for proliferation or mitotic damage. So that's possible. And in Sparkline, because we do not have a timeline data, that's why we cannot show it now. But if we had that value, we could have actually shown it as a line plot, as one of the values. So that's this one. Last, very interesting and very useful for you guys is, because ultimately we all do the comparison, right? Comparison amongst the different groups. So how do you do this? And maybe before that, I would tell you how to, I think, which is a good example for this one. Let's say, because the box plot, that's basically remaining. Oh, by the way, we haven't seen actually the heat map, right? Or this tree map. Okay, there is a possibility of actually plotting the tree map also. So let's go to tree map. Tree map, yeah. In tree map is based on the hierarchy, right? And you have to decide on what criteria you want to decide this tree map. On what criteria you have to decide the hierarchy of this tree map, right? So now I have decided based on gene, right? So it's just giving me, okay, this is empty, which is basically there is nothing is happening within this particular well, and then this the control, negative, and the size is the sum of 53 bp1, but I would like to go for the average. Okay, now they are actually similar in size because they are more likely, because it's average value. Because if we go for sum, it means that there are more wells of the negative control, because for every plate there is a negative control. That's why the number of cells are more negative control, and that's why you would see the more width of this one. But if you go for average, then you see equally likely. But the color, you want to give the color based on gamma H2X. Oh, sorry, the gamma H2X votes on the average value. Now the things have changed. So basically, maximum is this one, which is a dark color. So there is, so this guy, this gene has more gamma H2X, and the size is almost equal for 50 bp1, but there's something gamma H2X is more prominent for this particular gene. This is what it tells you. Yeah. So you can basically select it, and then, as I said before, you can actually right click here and say, okay, create details visualization, and maybe I want to see it as a scatter plot, right? So what the detail visualization is that once I select this, something will display here. Marker by, let's say, marker by siRNA, color by anything, that's fine. And here I'll go for EDU. Right click. Labels. I would tell, okay, what kind of siRNA it is. Yeah, that's basically control positive. That's this one. There are two siRNAs basically. This is very interesting, isn't it? And you can have as multiple details in visualization as you want. So you can have multiple such plots. So I can just go here, right click and say details, create detail visualization, but the pie chart. And in the pie chart, I would say, okay, I want to put this pie chart here. I would say the pie chart size by, let's see what happens. Then I will select this one. Of course, there's nothing here, right? But let's say if you select multiple such stuff, and if you select this one, there's a bunch of this. So basically, details on visualization with the different levels. So you select, okay, maybe there's something interesting for these guys because they're very dark. So there's a gamma H2X is more prominent. So put control key over here, control. And these are the sRNA for this particular gene. And maybe within this two criteria, which is EDU cell proliferation and the 15th B1, I'm interested in these sRNA which are more proliferating. What's their contribution based on some other criteria? That's it. Yeah. So that's sector size by which one we haven't considered here, gamma H2X. That's it. So there is two information and then this third information. Okay. Okay. That's all about this. The tree map. Yeah. Let's go for heat map. In the heat map, you should have the row and columns. That's basically heat map. Right? So if you go to column here, that's basically this one. But I do not like the heat map of this one. This is what basically it is. So that's a row and column, but based on what? We haven't given this criteria. Properties. Keep the color based on H2X. So that's basically right. So for first row and the average column, this is the number. So this doesn't reflect what we are actually doing in a microscopy. So I have found another way of having the same heat map but based on scatter plot. So you go to scatter plot, you say row here, column here, then go to row. This makes sense because this is 96 well right now, right? And then maybe this is not also interesting because it's like there is a space in between these two guys. So just right click, go to properties, formatting. There are so many stuff that I don't remember, but there is something that you say size, shape, type markers, like this. And maybe you're not interested in the genes, but you're interested in, okay, I want to see the plate heat map of the EDU proliferation. Okay. So this is basically as if you're actually looking at the microscope, the plate view of the microscope. Rows and columns stratified by this one. And you can change this color criteria. And again, the same thing as a details visualization. So you can go for these particular guys, which are very interesting. Wells, and then right click and say, okay, create details visualization and do this. Okay. This is all about the heat map. So we have seen all the modules by which you can visualize and analyze your data. The one is remaining that's called as a box plot. Okay. Before that, you can also have a text area. So if you want to write some kind of, if you want to sharing this information, or if you're sharing this visualization to your colleague, then if you want to write some information that this is what you should look at, the page number 1 is this one, that meta information you can write it. So go here and say edit text and write this is my page and something like that. Now you can change the font size. Okay, that will remain there. Yes, you can change it. By the way, you can change the background color, okay? So by default, it's a light color, but you can go to dark color. Everything is fine. just delete this text stuff. Let's go to box plot. So if I use a box plot here, now here is bit kind of a statistics, right? So box plot is x-axis. You are just giving the groups that you want to compare. So right now I want to compare genes basically with respect to each other. But on what criteria? On criteria based on, let's say, why not 53BP1, right? Which one are more mitotic damage marker? And if you see here that now, do you know what is box plot, right? So the box plot, I'm just reminded. So the box plot is, if you see this line over here, that's a median value, okay? And the upper age here is third quartile. The lower age is the first quartile. Now what is quartile? If you, let's say if you have 100 cells, every cell will give you some value of 53BP1. You sort this 100 numbers from increasing order, minimum to maximum. You just go to the middle position. That would be, let's say if you have 101 cells, then the middle position would be 15th one. Then you just go there and look at it, what the 53BP1 value is. That is nothing but the median, right? Similarly, if you just go to the 1/4 of the position, which would be the 25th percentile, that's the first quartile. Okay, so that's the first quartile, third quartile, and the median is the 50th quartile. I mean the second quartile, basically. Yeah, 50th percentile and the second quartile. That's a median value. So in the box plot, the reason why the box plot is important to compare is that it is non-parametric, right? Because you're comparing the median value. You're not comparing the average value. I mean, that is possible. I can show you how to do it. But before that, maybe because this is basically too much of complication because there's so many genes are there. So what I would do, I would go back and say there's a filter here. Filter. If you click this filter stuff, I can reduce my genes. So maybe I'm not interested in all the genes that I want to compare, but maybe some of the genes. So if you just click here, control, put control. I think this should be now. Now it looks good actually, visualization. Imagine that your interest is only to compare these guys, these genes with each other with respect to 53BP1 foci. Then these are the median values. And just by looking at this box plot, I would say actually this gene CALHM3 has more 53BP1 kind of feature likelihood. So there is something interesting for this particular one. Now how different they are from the control. So let's say my control is empty, which is control basically, right? It's not negative or positive, but it's just a control, or BRCA1, which is also control that way. So they are basically more prominent than BRCA1. But how much prominent? I can't say. That's basically p-value, right? But just by looking at the median, I can say actually they are different. Just to look at, then also I'm interested in knowing the confidence interval, right? Because you know the rule of thumb that as long as the confidence interval of the two groups are not overlapping, they are statistically different for sure. You don't need any p-value or stuff like that. It's very clear. So right click, properties, go to appearance. In appearance you say that I want to know the show 95% confidence interval of mean. That's the interval of mean, by the way. Okay. Also. And because our background is now black, I would change it to light. And you see this black stuff, that line, the vertical line, that's basically confidence interval. And you can see this confidence interval of this guy is not actually overlapping with respect to BRCA1. So I would say completely that this is statistically different with respect to 53BP1 with respect to BRCA1. And likewise you can compare. But Spotfire can also give you this already. So you don't have to visually look at it, but go to properties, go to appearance and say show comparison circle. And in comparison circle you have to give the level of significance. Because every p-value is dependent on the level of significance. So by default, everybody has accepted in scientific community that 0.05. If the p-value is less than that, it is acceptable. Well, there is also debate on this these days, right? One has to reduce it to 0.001, 0.05. But that you can change also here. So I would go for, and I would give to, let's say, I would always go for 0.001. That's always one should try. But then nothing is significant, right? Then in this case, you have to improvise your sample size. But apart from this, now it looks okay because now this circle is not overlapping with this circle. So meaning that these two guys has to be different statistically. So let's say control, click here, these two guys are for sure. Okay. So, this tells, as I said, I think the statistical lectures, that this is just the maximum value and this is a minimum value, and anything, this is a viscous basically, viscous because that's a viscous plot, that's a box plot. Anything beyond this viscous is considered as an outlier, right? So there's nothing, there's no outlying it. That's why you see 0, 0, 0 everywhere. With this, maybe you're interested also in not knowing the median value, but also you want to plot the average value. So for that, just go and say statistical table. You just say I want to actually visualize the average value. But this is for getting the numbers in the down. These are the numerical values, right? But maybe you're also interested in the reference point. So right click, properties, go to reference points, and here maybe you would like also to show the average values. But average value by, yeah, I think this would be okay. Dashed line, and if you want to give some different color, that also. So you see, you can plot everything in one particular plot. You have average value, you have median value, you have confidence interval, you have interquartile range, everything. So this is why I always refer to the users to show to their plot. Okay, and that's the average value. Good. This is all about the box plot. Any question with this box plot stuff?
Would you recommend Spotfire for box plots or different software?
No, I think Spotfire would be good, because unless you want to show it in a different box plot, because there are different ways of plotting box plot. There are multiple ways, right? Tukey's ways or the viscous ways. There are so many other algorithms, but I think this is the best and commonly used algorithm. This is a box viscous plot. So viscous is a method by which you can plot. Otherwise, you can plot the notched box plot also. Right. Because if you use a graph pad, normally in the biology people use this notch basically. So they show the distributions plots as a box plot. Right. That is also one way, but it's just a representation, right? I think for me it is important to show all information in one plot, which is actually in this case, right? You have a confidence interval, you have mean, median, interquartile range. That's important. Here you cannot show it as individual dots. Okay, but what you can do is you can go to properties, appearance, and go show the distribution. You can plot a distribution actually. This should be self-explanatory. Actually, this is the best way to explain, because just by looking at this data, I would say that the distribution of maybe for this guy here, it is not normal. So there is no point in showing t-test, p-value, which normally people does.
So that's right from the average and the median, right?
Yes, exactly. Because they're not overlapping. So basically in this case, I would say yes, kind of similar, because average and medians are overlapping almost. That's fine. But I think, see, this is the best. I think everybody should do this, right? Because then a reviewer would come to know what kind of a distribution it has. He need not to know about the standard deviation, standard error of mean or confidence interval. You already have the confidence interval, but everything is there. Right? By the way, as I said before, also there is also possibility of showing the scatter plot, as I said before, right? So you just go to what was it? This random stuff, right? 53 BP1. If you select it. Properties. Really smart. That's basically it. It's kind of a box plot, but now you're showing all the individual dots basically. But here also, as I said before, you can also show and say the horizontal line. You can say I want to draw lines. Horizontal line, aggregate value. Maybe you can use the lower 90% confidence interval. So here I actually can show confidence interval or the median value. Let's go for median value. That's a median value. Okay. And also similarly, you can show with the different lines confidence interval. So it's kind of,
you don't only add the whiskers.
No, exactly. Because this is not actually the viscous plot, right? This is basically a scatter plot, but we are trying to tweaking this. Okay. But this should be enough. I mean, this is normally people without distribution, showing distribution. This is common practice, especially if you read this Nature guidelines for publication. I think they would prefer box plot to be shown users. Okay.
So if you have 3 experiments, that's the same thing. How would you present the data?
That's basically a consistency with the that's a reproducibility, right? So then you should show the standard error of mean within the replicates. Yeah. But that's basically statistical part. I mean, that's that we can cover later also. I already covered actually.
So one experiment is 2,000 cells.
I know. Yes.
It's used to be this. They just saw that and then said we need 200 times. You just don't need that.
Exactly. I mean, especially those who are working with embryo stuff. This is everyone should follow this guideline, right? Because if you're comparing one embryo with another embryo, you cannot just pull all the cells within the embryo and then compare it, because within one embryo you might have hundreds and thousands of cells, and because you have hundreds and thousands of cells, because of this huge sample size, even if it is not statistically reality significantly different, but because your sample size is so huge, you will get p-value significant. But this is what one should avoid doing.
You're saying something for the replicates?
Yes. Yeah. Because this is interesting, right? That how reproducible is your experiment than showing the standard error of mean for the individual experiment. Okay. So this is about the box plot. I think we covered everything actually. We covered everything. Now only one thing is remaining about the labeling, which is also important, right? So let's say let's say that these 3 genes, you know a priori that they belongs to one particular category. Let's say, I don't know, you guys tell me, positive or negative, something like that, with some which basically responsible for happening something positive and negative in biology. That's it. So I select. So before that, okay, before that you have to click on the tags. So what I'm doing now, I'm basically making an artificial labels, which is not there in the data itself, but I'm making a category from the user, by the user. So if I click on this one, tags, go to this main tag, you have to give the name, let's say comparison, knockout, and let's say one time. It's completely fabricated one, but just say knockout, that's knockout, and then one time. Okay. Now, right now there is a 0 candidates are assigned to these groups. I can actually select from this one. You need not to be on the box plot, okay, by the way. You can be on any of these. Say, I think this is more important. I'm going to do it this way. So you select all the cells from this particular genes, select it, and then you go to, okay, but first you have to create on the same page the labels, okay? Now it is not there. So you go there, where we have created, where was? Here. But you understand my point, right? It need not to be the same visualization, but you just select, let's say these 3 guys. Right click and say attach tag to the marked rows. So all these three genes will be considered as a knockout, and these three will be considered as wild type. And likewise, you can create multiple groups and such multiple labels. Now what we will do, we will go back, and instead of genes now, we will go comparison KO to wild type. You're gonna see only two explode because it is pooled basically. Right? And then you can compare and see, okay, they are statistically different actually if you combine them. And likewise, you can create multiple such labels. This is one part. Very important is that because you remember that we have selected few of the genes, we haven't selected all of them. But if I select and if I go to the other page and if I try to use now, let's say, scatter plot based on particular genes here, I am only restricted to that particular gene that I have selected in the previous page, which is actually not good. What if I want to do two separate filters for two separate pages? So question is it possible to have a multiple filtering scheme for two different pages? Right. Maybe I am processing this data. I have certain criteria for the filters, and maybe you have certain criteria for the filters. So how to create such criteria? Because right now, if I want to, I cannot see now. I can only see the selected one, but maybe I want to do some other selection. So for that, you just go to edit, document properties, go to filtering scheme, show filtering scheme menu into product bar, go to new, and say user 1, user 2. Say okay, that's it. Now you just go here. That is user 1 setting, and you can select now the genes that you are interested in. And if you go to this page, say user 2, you got it? And then you can completely select the other criteria for selection, maybe this one. So there are two independent now filtering scheme. You can also right click in this page and say rename page, give some meaningful information for that particular visualization so that later you come to know. So I think we have covered most of the, I think all of them, right? Because nothing is remained. Oh yeah, one thing is remained is that if you have a main object and sub object and how to combine them together.
I have to go.
How to combine them together. Okay. So we have seen this example of a foci data. So it's a main file and this is the foci file. So all the features will be extracted from foci and the main object. How to combine them together. So let's see this. So go to close here. First, drag and drop the sub object because we want number of rows to be the same for the sub object, right? It is okay if the one foci has a multiple main object criteria, which is okay. So I'm just drag drop the sub object, which is basically foci related features. Yeah, and then I go to insert, and what do I want to insert? I want to concatenate rows or column. Of course, columns, right? Because foci features, and then besides foci feature, every foci will have a common main object feature. Like if that foci belongs to, let's say, Tabi intensity, then if there are 15 foci, then all 15 foci will have the same number, which is okay for us. Insert columns. And then go to this location. File. Go to that location. Get this main value. Okay. But there is a point here. It won't. Spotfire won't do this automatically. There has to be a key between these two tables, right? Common key, which would tell this row belongs to that row. And whenever you extract from Scanner or from Imaris or any other software, they have this common key. So for example, in Imaris, if you have a foci vesicles, if you're tracking the vesicles, every vesicle will have a cell ID. Okay? And if you have an information for the cell, every cell will have a cell ID, of course. Right? Then basically the common key here is a cell ID. Right? So in this case also there is a common key called as a object ID. Okay? So in this case, you first import everything, go next, and then here it is asking us which key you want to assign. So my original data was foci data. So foci data parent object ID, that's my key, and object ID, that's my key from the main data. Max selected, go next. Now it is asking me, okay, I have all my data from the sub object, which is a foci object. In addition to that, which column do you want to actually concatenate from the main object? That's my choice. I can say all of them, or I can select a few of them. Maybe I'm interested in total Dabi intensity, mean intensity, circularity factor, perimeter, and also the PMR, foci count. Okay.
Why don't you just do that yourself in Excel, for example?
Yeah, you can do it in Excel. That's basically a join operation. You can do inner join, outer join, but my point is that you don't have to use an Excel, right? Because it's just that telling you that it is possible to do it everything in Spotfire side, right? So, and then go to finish. That's it. So if you see now the data, click on the tables, you will see the extended columns. It's here, right? So every object ID is basically, this is the foci, and for every foci you have count. See, this is 15. So there are basically 15 such. Yeah, exactly. Yes. So there has to be 15 somewhere here. Yeah. Yeah, so I'm sorting, but there has to be because we have used the same key. Okay, it's just that we have to sort it. Parent object. Let's go for this one. That's parent object. So 0, 0. There has to be 5. Anyways, but you understand this point, right? So there has to be a key, common key, which would join basically two columns, and then you can actually do everything now. Scatter plot, and then you can go to object ID, and here you can say count. Stratified by okay, so this is all about then. You can also calculate columns. So if there is an additional column that you want to, let's say for example, you want to calculate the average value of this total intensity of DAPI and the total background corrected DAPI. Just hypothetical situation. That's also possible. So you just come here, insert this column, and then say plus. Insert this one, plus it. Then do this. Do that. Okay. And then you say here that average and also see the GPU of this one. That's basically. And say okay. And this new column. So that's possible. You can also insert, and then you can also say calibrate column, predicted columns, transformation. That's also possible. So you can do normalization. You can normalize a particular vector, basically. So normalization. Let's say add. You can. These are the normalization that one can try on theta. So maybe I want to apply on total side beam intensity between 0 and 1, and it would be called as a normalized one. So add this. Let's see, okay. If you see now the table, you should see the normalized. See normalized? Yeah, there is normalized. That's it. And you can use that as a criteria to generate your plots. So I think we covered almost everything. Almost everything.
So if you have councils, often you think we're discussing this. Let's say that you do your experiment, you stress them or not stress them. 4 conditions.
Yeah, 4 conditions.
Control the side, right? Let's say it's 4 conditions. Then you have the second slide, which has the exact same conditions on a different side.
Right.
One thing that I have forgotten is how to import that one in, and that's the other way around. So when you import the data there, you can file one next to each other.
Yeah.
You import them as rows instead of columns.
Yeah, I got your point. Okay. Yeah. So imagine just the hypothetical situation. Now imagine that you have done this experiment and you have repeated this experiment. Let's say you have 3 replicants, and now we not going to have 3 replicants, but imagine there are 3 such files. Okay.
So 2 query genes and 1 is your control.
Yeah. Let's say if I do like this, do you agree with this one?
Yeah.
These 3 replicants. Now the question is, how do you take all these guys into one Spotfire and showing them this replicant 1, 2, 3, 4, right? So the way you do it is go to Spotfire again. I will open one more. Actually, this is very interesting because for this you have to use labels and also concatenate at the same time. So for this one, you open first this file, main file.
But if you work with class lines and cover slips along, this is probably the way to do it.
Yeah.
You can go up to 8, you can have 2 more than that. So
No.
you have to control stuff.
No.
6.
Yeah. But if you have many such replicates, then of course it's not feasible for you to do it like every time this thing.
Sure.
Then it's just a small script that you would write. Okay. And just come to me if you have many replicates. Because so far I think we don't do that high throughput in that way.
No, but it's also high throughput. You have a gene and then you just control it with 2 or 3 SRNAs.
Oh, yeah.
Compared to the
Right.
to the scramble side.
Yeah, exactly. So like I'm importing the first file now. And then go to this labels. And I would say, okay, this is my experiment number 1. I did this is my 1. That's it. Then here I would say, okay, that's the experiment that I would like to see. So this whole everything would be basically separate number 1, right? Then go insert. This would be rows because the columns are same, right? These guys, that's an application. Go there, files, copy, go next, match all possible, which we know, right? And that's it. So this untag would be the number 2. Insert rows. Yeah, you got the idea, right? There is no point in showing the box plot now because this is just real application.
Kind of files to export. Like
Oh yeah, exactly. That's very important. So let's say you have done everything what you wanted as a visualization. This is very important. How to export this data, right? So let's say, for example, you want to discuss this in a group meeting or something like that. Then one thing Spotfire can give you is directly go to this tab over here. That's for PowerPoint export. If you click that, say entire page, that's your choice. You want to go for the active visualization or the active page, meaning that this complete page. So let's say active visualization. Because active visualization meaning that if within one page if there are multiple visualization, then it won't be there. So it's active visualization. Do you want to go for you want to display the visualization title? Yes, this title will be displayed, otherwise not. Filter settings, you want this to be displayed. Page titles, that's your choice, right? Then you would say new PowerPoint presentation, say export. Oh, I should have done editable so that you can also edit it, right? So then just open. This is open. Isn't it nice? But it's also possible that you just go and while exporting, you can say export as editable image, so that in the PowerPoint you can actually edit it all the box. You can ungroup it and then assemble according to your need. So if you go like this, by the way, you can actually ask them to present the second slide, export it. Now this is edited, right? Right click and say group, ungroup. That's fine, yes. It's ungrouping all the objects. The reason why it's taking too long is because microscope. Yeah. Oh, sorry, I should have done this. But you understand now. Okay. Okay.
Do you also use the themes?
Yes. So if you just go here and say export to PDF, HTML, or image. So in an image you can have PNG, GFP, by the way, did I say that you can export as an XLS file again? Let's say if you do some kind of a calculated columns and stuff like that, or maybe this one. This is very interesting. I said before, right? So yeah, you can have this multiple stuff for different gene, for example, here. You got my point. And then you can just go here, export it, export data to file. Go to data table, all rows. Say OK, and then save it. That will be an XLS file. So right from visualization to data analysis, it does. And there are so many other advanced data analysis you can do, like for example hierarchy, clustering, regression modeling, all stuff. And then you can also write R script if you're proficient in R. That's also possible. Okay. I don't know, there are so many YouTube videos, tutorials on this one, because this Spotfire is actually booming up now. So as I said before, I think most of the pharmaceutical industries. And you being a biologist, if you have this ability to explore the data, I think that's the best career option one can imagine.
For the same cycle, most people are doing both dumping and cycling at the same time, right?
Yes.
That's what we decided to do. The scatter plot, or the one that have dumping in intensity or whatever, and then the other one that have cycling.
That's a line plot basically.
Okay. So line just selected the relation there. So when we say G1 will be the cycling negative plus the dumping least, so it would be like the left plot.
So basically, x-axis would be your time, and y-axis would be the ratio.
No, it would be time is not linear because it's just fixed cells.
Oh, okay. So you're talking about fixed cells.
Yeah.
The data.
No, it's not live.
Okay.
Okay. So they would select the double ones. Pretty much what you were doing with the meaning for the dumping.
Right.
Instead of having dumping to dumping, you have dumping to the second one.
Second one, exactly.
And you select ones, and you have to tag them afterwards. elements, right?
Yes.
So you tag them, create a new one, and then you say just measure whatever you want.
Right, exactly. So in that case, I would create first a mask, which is around nucleus, and I will just expand it for a few diameter, few micrometer pixels, and then I will make basically two sub-objects. One is within the nucleus, and one is just outside nucleus, right? Okay. And then I calculate the mean intensity of cyclin B1, and I see.
No, cyclin A isn't better because it's just nucleus.
Oh, okay. Oh, okay. Then just
outside.
compute the mean value of cyclin B1, so cyclin A.
To the
DAPI, and then you have DAPI here, and then cyclin, and this.
So you have actually two these things, the thing that you just described.
Yeah, the profile.
Yeah.
But for profile, you have to have the time axis.
Yeah.
The profile. Yeah. I mean, if you have enough such continuous variable, then that can also be considered as a time.
But most people select on the DAPI only, or do they do additional stuff?
I mean, the project that I was handling with the second floor CPR, that was the live cell, and that was these two channels that were cyclin B1. But that's completely different, right? Because they wanted to know the transition between these two. Yeah, exactly. Okay.
Thanks a lot.
So for example, Feynman, Kai, and the rest, I think they all select primarily on DAPI.